Xstrahl hosted a virtual meeting, Advancements in Translational Radiation Research, featuring a panel of leading radiation researchers highlighting translational research done over the last 10 years using the Small Animal Radiation Research platform. The meeting featured 6 leading radiation researchers who highlighted some of their research outcomes using SARRP. Dr. Anthony Chalmers, Chair of Clinical Oncology of the Institute of Cancer Science at the University of Glasgow, summarizes research done to study the immune response to radiotherapy treating rectal cancer.
Some newer work looking at orthotopic transplant models of rectal cancer. So these studies are to investigate immune responses to radiation in rectal cancer. And what we've done, collaboration with Owen Sansom's team in the Beatson, is to develop syngeneic tumours derived from murine tumours which have been cultured as organoids and then injected in immunocompetent mice using a colonoscopy to inject these organoids submucosally so that we have the tumours growing in the correct anatomical location. The organoids have been engineered to express combinations of common colorectal cancer driver mutations as shown here, and the histology of these is very reflective of the human disease. We've been able to demonstrate an induction of DNA damage in these tumours, and as I've mentioned, we want to use this to really study the immune response to radiation in these immunocompetent mice. So a very simple system is just again to use a single superior beam. In some of our mice we can very clearly see the rectum because it contains gas, but in others, such as this one, you can't. So this is still fairly speculative isocenter placement and planning. We are endeavouring to improve this with the use of enemas, either of air or perhaps a contrast agent to improve organ delineation here. And then we're also looking at the use of arcs to perhaps spare some of the normal tissues, because obviously when we're looking at the immune response, the irradiation of the adjacent normal tissues can play a key role in that. So this first attempt with this arc setup, we showed that we can very nicely exclude air from the volume, but that's about it. So we're now testing a different setup, and in this one we're specifically aiming to exclude the spine and the bone marrow from the volume, and here I think we've shown that we're capable of doing that with this beam setup.
For more information on the Small Animal Radiation Research platform, or to learn how SARRP can help you with your research needs, visit xstrahl.com/SARRP.